Okay, so welcome back to this next video in which we are discussing Parkinson's disease and anti-Parkinson drugs. Okay, so in the previous video what we discussed was the symptoms of Parkinson's disease, both the motor symptoms and the non-motor symptoms. Okay, and we also discussed that the fundamental process that underlies Parkinson's disease is neurodegeneration. Okay, neurons are going to die all over the brain. Okay, and one of the key areas of neurons that die, as far as the symptoms are concerned, is the substantia nigra pars compacta, as we'll come on to. Okay, right, so we want to have a look at what is the neurodegenerative process underlying Parkinson's disease. Now, as far as finding out about the neurodegenerative process underlying Parkinson's disease was concerned, what has been tremendously helpful is that there are very, very rare cases of hereditary Parkinson's disease, okay, where the members of families in which there is this hereditary Parkinson's disease being passed along the generations, those people in these families can develop Parkinson's disease much, much earlier than a sporadic case of Parkinson's disease would occur. Okay, and I want to stress how rare these hereditary forms of Parkinson's disease are. They are very rare within the population. Okay, so what is interesting then about sporadic and hereditary Parkinson's disease, and most Parkinson's disease is sporadic, okay, is that we think the pathological process that is occurring in both of them is the same, okay, it's just going to be caused by different things, okay, and that's interesting. If you contrast that to, for instance, breast cancer, where the hereditary form of breast cancer is completely different from the sporadic form of breast cancer, okay, that's nice as far as learning about it is concerned. Okay, right, so this protein then, alpha-synuclein, is going to be at the centre of Parkinson's disease. Now the first thing that I have to say about this is very disappointing. We don't know the function. Okay, so the function of alpha-synuclein protein is not known. So function not known. So all I can do really for the protein alpha-synuclein is represent it as a box with alpha-synuclein written in it. I'll colour it in to make it look slightly more interesting. So here is alpha-synuclein here. Okay, right. Now this is the protein which we think aggregates in Parkinson's disease. Okay, well in fact we know it aggregates in Parkinson's disease. So let's have a look at the certain hereditary forms of Parkinson's disease which are going to involve mutations in alpha-synuclein. Okay, so two mutations in alpha-synuclein that have been found are the A53T mutation, okay, and also the A30P mutation. Now let me just take this as an opportunity to make sure that everyone understands how to read this mutation naming system. Okay, basically the first letter here in both cases is an A. Now what that first letter tells you is what the amino acid was. Okay, so in a mutation what's going to occur is one of the amino acids, well, these mutations in alpha-synuclein are substitution mutations. Okay, you're going to turn one amino acid into another amino acid. Okay, and that's what this notation here is telling you. It's the first letter tells you the first amino acid that was originally there. The last letter tells you what amino acid is going to be there after the mutation has occurred, and the number in the middle tells you which amino acid we're looking at. So this tells us that we're looking at the 53rd amino acid within alpha-synuclein. So it is fundamentally a polypeptide. So if I wanted, I could represent it actually as a polypeptide. So maybe I could represent it something like this. Okay, and this line would represent the polymer of amino acids. Now if you counted along to the 53rd amino acid, normally it should be an alanine, okay, at position 53. Okay, it's now going to be mutated to whatever T stands for. So to be able to read this nomenclature, you need to know single letter amino acid code. Now generally, single letter amino acid code is very easy. Generally, the letter used is just the first letter of the name of the amino acid. So for instance, A is alanine. Okay, so let me just remind you of the structure of an alanine amino acid. Okay, so here is the amino group, and I'll draw it as though it's a residue, so I'll draw it as though it's bound within the polypeptide. Here is the alpha carbon with a hydrogen coming off it, and here is the carboxylic acid group here. Okay, now the R group of alanine is just a methyl group like so. Okay, so this is the amino acid that you should have at both position 53 and also position 30, so I'll put that here. So we've got these two alanines. Okay, now in one of these mutations, the alanine at position 53 is going to be changed to a T. Now T is the single letter amino acid code for threonine. Okay, so once again, I'll show you the structure of threonine. So here is the amino group. Okay, here's the alpha carbon. Here's the carboxylic acid group. So that again is just the core amino acid structure. And then threonine contains, well, the R group of threonine is a carbon with an alcohol group coming off it, a hydrogen coming off it, and then a methyl group coming off it as well. Okay, so this is the structure then of threonine. Okay, so in this mutation of alpha-synuclein, all you're going to change is this alanine at position 53, and you're going to change it to a threonine. Okay, right, at the second type of mutation that was found in different families, okay, the alanine at position 30 is what's going to be changed, and it's going to be changed to a P. Now, P is the single letter amino acid code for proline. Okay, so let me show you the structure of proline residue then. Whoops, I nearly went wrong there. I can save it, however. Okay, proline is an unusual amino acid, okay, because it does not have the hydrogen coming off the nitrogen in the residue like so. Okay, because remember, the R group of proline involves the nitrogen. Okay, so here is the alpha carbon, here is the carboxylic acid group. We'll put the hydrogen on this side, and now we're going to have this 5-membered ring here as the R group of proline. And each one of these carbons is then going to contain 2 hydrogens coming off it. Okay, like so. Right, so that's the R group of proline. So we're going to change an alanine at position 30 to a proline. Okay, right, and that's the second mutation that has been found in alpha-synuclein. Okay, right, so these are some of these mutations that are found in these families which have this very rare hereditary Parkinson disease running through them. Okay, right. Generally, having a mutation in alpha-synuclein predisposes you to early onset Parkinson's disease, but it doesn't quite predispose you to the point of getting Parkinson's disease at around 3. Okay? Instead, what will happen is generally you get Parkinson's disease at around age 45 rather than age 60. Okay, so it's just moved it forward by 15 years. Okay, now why does having a mutation in alpha-synuclein cause Parkinson's disease? Well, what seems to happen in Parkinson's disease is that alpha-synuclein is going to aggregate. Now, what we think is that if you get mutations in alpha-synuclein, okay, such as one of these 2 mutations here, what it's going to cause alpha-synuclein to misfold. Okay, so these mutations, we believe, will now cause alpha-synuclein's tertiary structure to change, okay, i.e. it will misfold. It's now going to fold up in a totally different way. So how can I show this? So maybe now it'll be folded up something like this, maybe. So it's looking completely different to what I've drawn over here. Okay, and I think I'll colour in alpha-synuclein. Once again, I'll colour it in green. Okay, so here is alpha-synuclein, here in green. Okay, and now it's misfolded in this new way. Okay, and now what's proposed to happen is that this misfolded alpha-synuclein is now going to aggregate into fibrils. Okay, so what's going to happen now is it's going to start oligomerizing. Okay, so if I now represent one alpha-synuclein protein just as a blob, what's going to happen is multiple of them are going to start aggregating together inside the cytoplasm of cells. Okay, and they're going to start forming little fibers. Okay, and I don't know if the fibers are actually 2 alpha-synucleins thick like this. Okay, the principle is just that they're aggregating together into a fibre-like structure. Okay, and these tiny little fibres that you're going to start building inside cells are known as fibrils. Okay, so you're going to start forming these fibrils of misfolded alpha-synuclein proteins. Okay, and then the understanding is that these fibrils of alpha-synuclein proteins are going to damage the cell in some way that's then going to lead to the cell dying. And it's not known how this accumulation of alpha-synuclein actually kills the cell. What is suspected, however, is that it's going to cause damage to the mitochondria in some way, and that is then going to cause the death of the cell. So it's going to cause mitochondrial dysfunction in some way. Okay, right. The next thing to say is that inside the cells that are of someone who is suffering from Parkinson's disease. So if you look inside neurons of someone suffering from Parkinson's disease, maybe they have hereditary Parkinson's disease, or if they have sporadic Parkinson's disease, it's actually in both of these cases, which is what's nice about Parkinson's disease as far as learning about it is concerned. You find structures known as Lewy bodies. Okay, so let me explain what a Lewy body is. Okay, so let me draw a neuron firstly. So here is my representation of a neuron. Here are the dendrites coming off the cell body here. Okay, like so. And then we'll have the axon here. Okay, and then we'll have the nucleus here. Okay, now a Lewy body is going to be a cytoplasmic inclusion, so it's going to be inside the cytoplasm of the cell. So let's put one here. Okay, and they look like this. Okay, they have two sections. They have a dense core at the centre, so that dark bit, I'll draw it outside as well so I can label it up. Okay, so it's just this sort of fried egg structure here. Okay, now the really dark portion at the centre is known as the dense core of the Lewy body. Okay, and the portion around the edge, the lighter portion, that's known as the halo of the halo of the Lewy body. Okay, right. So you find these Lewy bodies inside neurons inside the brains of people with Parkinson's disease, both hereditary and sporadic Parkinson's disease. Okay, now when you actually examine the Lewy bodies, you find that they contain a huge amount of alpha-synuclein. Okay. Now, you might wonder, well, are these things part of the pathology? Are these things the things that are killing the neurons? Okay, and the answer is we don't think that these are the things that are actually killing the neurons. Okay, but they're certainly part of the pathology. They're certainly part of the fact that this neuron has far too much alpha-synuclein. What we think is more likely to be true is that the Lewy bodies are effectively dustbins. They are places where the neuron is just dumping this alpha-synuclein. Okay, and we'll see in a moment that in order for alpha-synuclein to go into one of these Lewy bodies, it seems that it needs to be ubiquitinated. Okay, and we'll come on to that in a moment. Okay, right. So that's the Lewy bodies, okay, inside neurons. Okay, right. And then, as I've said, the hypothesis is that this accumulation of the alpha-synuclein inside the cytoplasm of these neurons somehow leads to mitochondrial dysfunction that then causes the neuron to die. Okay, so to summarize then, for these alpha-synuclein mutations, what happens is we think these mutations in alpha-synuclein cause the alpha-synuclein to misfold. Okay, then the alpha-synuclein proteins start to aggregate into fibrils. Okay, so you get far too much of the alpha-synuclein building up in these fibrils inside the neuron. Okay, the cell sort of starts dumping alpha-synuclein into these Lewy bodies, and we'll explore that more in a moment. Okay, and then somehow this build-up of alpha-synuclein then causes mitochondrial dysfunction is the current hypothesis. Okay, and we'll talk about why we think mitochondrial dysfunction is so important. We'll mention MPTP later on. Okay, so mitochondrial dysfunction. So remember, the mitochondria are the energy houses of a cell. Okay, they produce the energy, they produce ATP. Without them, if they're not functioning properly, the cell has no ATP, and that's the biological energy currency. That's what drives most of the chemical reactions that occur in the cytoplasm of the cell. So without that, the cells are going to die. Okay, right, so mitochondrial dysfunction. Okay, so now to transfer it over to the sporadic case, because that's hereditary. That's lovely to know about, but this is really, really rare, whereas the sporadic case is much, much more common. Okay, so to move over to the sporadic case of Parkinson's disease, how does alpha-synuclein play a role there? Well, you don't have a germline mutation in alpha-synuclein. Okay, that doesn't mean that a cell within the brain hasn't got a mutation in alpha-synuclein and then has produced this misfolded alpha-synuclein that could have spread to other cells surrounding it potentially. Okay, but so far we have not found mutations in alpha-synuclein in the neurons in the brains of people with sporadic Parkinson's disease. Okay, so instead what we think at the moment is potentially some environmental factor, so potentially exposure to certain chemicals, and the one that everyone's pointing at the moment is certain chemicals that are used as pesticides, okay, especially chemicals that are sprayed onto crop fields, okay. Now the reason that people are pointing at those is because certain of them are mitochondrial toxins, okay, so that's all linking in now, and also the fact that Parkinson's disease is generally actually more common in people living in rural areas, okay, where they are potentially more exposed to pesticides used on crop fields. Okay, so that's what's kind of pointing to potentially herbicides and pesticides, particularly pesticides, being potentially the cause of sporadic cases of Parkinson's disease. And the understanding then is that some sort of environmental factor, maybe one of these chemicals, is then going to cause the alpha-synuclein to misfold. Okay, so rather than it being a mutation that causes alpha-synuclein to misfold, it's some sort of environmental factor. Okay, so in the sporadic cases it will be an environmental factor, and then of course what you're going to get is gradually you're going to get more and more of this misfolded alpha-synuclein accumulating in your neurons, forming the fibrils, just as in the sporadic case, just as in the hereditary cases. You're also going to get the formation of these Lewy bodies. Okay, so once again the cell starts dumping the misfolded alpha-synuclein into these Lewy bodies, and eventually the buildup of the alpha-synuclein becomes so bad that you get mitochondrial function collapsing, and then the neuron will die. Okay, and that could underlie sporadic Parkinson's disease. Okay, right. So we're now going to look at another type of hereditary Parkinson's disease. Okay, because it isn't just mutations in alpha-synuclein that have been found to cause hereditary Parkinson's disease. There are mutations in other proteins that have been found to cause hereditary Parkinson's disease as well. And this is actually going to be extremely helpful because it's going to paint this picture of what is happening in Parkinson's disease for us even more, basically. It's going to add even more to the picture. Okay, so the next protein that we are going to talk about then is a protein known as Parkin. Okay, right, and it is loss of function mutations in Parkin that cause the hereditary form of Parkinson's disease. Okay, so in the case of alpha-synuclein, these were both gain of function mutations, okay, because you got this mutation and then it caused the misfolding of the alpha-synuclein, and now the alpha-synuclein has gained the function that it can now aggregate into these fibrils. Okay, that meant that you only needed one gene to have got one of these mutations. Okay, so remember, we are diploid. We have two copies of genes for every protein. Okay, except for on the if you're a boy and you have the X and the Y chromosomes, but we will ignore that. Alpha-synuclein is an autosomal gene product. Okay, right. In the cases of these mutations, you only needed one of the genes to have one of those mutations, because you only needed one of the alpha-synucleins to gain this property that it can misfold and aggregate into fibrils to get the disease. Okay, in the case of Parkin, okay, which I know we haven't talked about what this is yet, but I'll tell you this much, that it is an enzyme, okay, and it's going to perform some function, okay, which we'll see in a moment. In the case of Parkin, in order for it to cause hereditary Parkinson's disease, you need loss of function in both genes. So both of the two genes for Parkin need to have suffered mutations that mean that they are no longer functional in order for this to actually cause hereditary Parkinson's disease. And this can cause really severe hereditary Parkinson's disease. So early onset that it can be as early as 3 years. Okay, so you can get Parkinson's disease when you are 3 if you have 2 loss of function mutations in the Parkin genes. Okay, so this is clearly very, very important. Okay, so what does Parkin actually do then? Well, as an enzyme, it is what is known as an E3 ubiquitin ligase enzyme. Okay, so what does that mean? Okay, well, if you do know what this means, I'll give you the punchline now. It's an E3 ubiquitin ligase enzyme that adds ubiquitin onto alpha-synuclein. Okay, what we're going to do, however, is we're going to go through what actually is an E3 ubiquitin ligase enzyme. Okay, and to do that, we need to discuss the ubiquitin proteasome pathway. Okay, whoops, missed out the I. Ubiquitin proteasome pathway, which is a way of degrading proteins. Okay, right. So I think we'll have a break here, and in the next video what we'll do is discuss this ubiquitin proteasome pathway. We'll discuss what E3 ubiquitin ligase enzymes are, and then we'll see how Parkin is going to do this for alpha-synuclein.